When exploring around trees, you may have noticed growths that look like these on a plant's leaves, fruits, stems, branches, or roots. These abnormal growths are called galls and are a result of an interaction between a gall-inducing insect, mite, bacteria, fungi, or virus with the plant. In Australia, a common gall-inducing insect is the Trichuligaster acacia longifolia wasp. The species gets its name as it targets Acacia longifolia, a native Australian species of tree. The wasp lays its eggs into the tissue of the acacia tree, redirecting the plant's cellular growth and development to form a gall that engulfs the egg. The larvae then hatches from the egg and lives in the gall, benefiting from protection from predators, a source of nourishment, and shelter from the elements. The larvae then matures until the adult is ready to emerge from the gall. There are limited studies into the effect of galls on the host plant. Some recent studies have suggested that galls have a neutral effect on the tree. However, it is previously assumed that the gall production comes at a cost to the acacia tree, and the relationship is parasitic. The gall-inducing wasp uses the tree's limited resources to redirect the plant's development to form a gall, leaving less potential resources the tree could have invested into the growth and reproductive success and fitness. To gain a better understanding of the interaction, we set out to investigate how the presence of galls affect the growth and fitness of the Acacia longifolia trees. To do this, we measured indicators of these two factors, including the length of leaves, the percentage of herbivory on the leaves, the amount of bean pods on the branch, and the amount of damage the bean pods had. We predicted that the presence of galls on a tree would result in a negative impact on its growth and fitness. We tested our hypothesis by surveying several locations around Smith Lake, including Myler Lake National Park, Marine Campground, and Horse Point Road near the UNSW Smith Lake Field Station. Overall, collecting data from 30 trees. The trail at each of the study sites acted as transects, and the first 10 trees within 2 meter distance of the transect were surveyed. To help reduce bias, a random number generator was used to select a branch. For leaf length, the length of the first 10 leaves from the branch's base were measured using a ruler. Using the same 10 leaves that were measured for length, the percentage of herbivory was estimated, based on the total amount of leaf that was eaten. These estimates were made after training using the Zacks Herbivory Trainer app. The total number of bean pods on the branch were counted. Then the first 5 beans from the base of the branch were measured and the condition was recorded based on a scale created of 3 different categories: no damage, low damage, and high damage. The size of the galls on the selected branch were measured as well using a caliper. After collecting our data, we analyzed it using statistical tests known as t-tests and linear regressions, ultimately finding that there was no evidence that the galls had a negative effect on the plant's growth or fitness. Therefore, there is no evidence that the interaction comes at a cost to the tree, or that the relationship is parasitic. The insignificant results could be linked to potential confounding variables. This includes the height and mass of the tree and the distance of the tree from the edge of disturbance and habitat fragmentation, as well as the stage of development the tree was in, as younger trees are more susceptible to the growth of galls. Further research that includes these variables is beneficial, as these wasps are used as forms of biocontrol in countries where Acacia longifolia is invasive.